Hello, everyone. First of all, I would like to thank Mark and Martin for organizing this conference about the conservation of swallowtail butterflies and the given opportunity to present our preliminary work on the Queen Alexandra birdwing butterfly. So today I'm going to present a genomic analysis of this species, Ornithoptera alexandrae. This is a work led by my PhD student, Eliette Rebou, with the help of Emmanuel Chevalier and Marika Tulak for the molecular sequencing, and Benoit Nabols for the genomic analysis. Also, we were assisted on the field by Darren Bito and Conwell Nukara. So the Queen Alexandra's birdwing butterfly, abbreviated QUAB here for practical purpose, is famously known for both its beauty and size up to 30 centimeters. Being the largest world's butterfly, this species has attracted quite some attention since its capture and description in Papua New Guinea. The crab is a micro-endemic species, which is restrained to a small area of Papua New Guinea, north of Port Moresby, in the Oro province. It flies around the town Pampondeta, which is a lowland rainforest, but also in the adjacent Managalas Plateau, south of Pampondeta. It flies from 0 to 850 meters maximum, and the species is composed of two main populations, with no sightings in between, according to available data and personal communications. There is a mountain range separating the two populations, bounded in the west by the Mount Lamington and going eastward through a mountain culminating up to more than 2,000 meters. The Sibian Mountains limit the southern edge of the plateau population. Interestingly, the quab populations are not only geographically isolated, but also occur on different soil types. The two lowland population, the lowland population here in the red circle one lives on soils of quaternary sediments, while the island population here in the red circle 2 lives on volcanic and plutonic rocks of older ages. The quab feeds on the genus Aristolochae, and particularly the species Dielsiana, at least in the lowland population. Based on external morphology, there is high uncertainty about the species status or identification of the plant in the population of the Managalas Plateau. We will come back to this point later. Most importantly, the quab is considered as an endangered species by the IUCN Red List. It is so since the book of Mark Collins and Michael Morris in 1985. Since then, we have gleaned additional information, especially thanks to the work of Michael Parsons and Chris Mercer, for instance. So being restricted to an area of 2,800 to 8,700 kilometers squared, the species faces important threats, mostly due to habitat destruction rather than illegal collecting. However, we lack accurate information on populations size and trends. Obviously, this is difficult to directly count the number of mature specimens like we can do in vertebrate species, for instance. And the number of mature individuals is also called effective population size in genetics. And so there are alternative ways to estimate the effective population size using genomic data. And this can help us to remedy to this lack of knowledge and can inform us on the genetic diversity and assist the conservation strategy. Before, it is important to remind that the quab belongs to swallowtail butterflies, Papilionidae, which is a well-known family of butterflies, including 32 genera and about 620 species worldwide, which all originated about 70 million years ago. There are between 41 and 67 species threatened or near threatened to extinction, and so studying the quab can help us understanding population trends and how to mitigate threats for these species. Interestingly, caterpillars of swallowtails feed on toxic flowering plants, in particular the highly toxic plant family Aristolochaceae, as for the quab, from which they sequester the toxin into their bodies. In return, become toxic too. Based on ecological and phylogenetic data, we know there is a very high level of host plant specificity across the swallowtail tree of life. This host specificity is recognized as a factor of intraspecific divergence, which can eventually lead to speciation. Indeed, experimental and molecular studies have revealed the presence of key genes to detoxify plant secondary compounds. Studying genomes can then help us to understand the genetic adaptations to host plants. Swallowtails have stimulated a lot of works, and many hypotheses have been proposed to explain how species originate. One of the most famous hypotheses is referred to as the escape and radiate model. The model makes the hypothesis that when insects acquire the ability to circumvent the chemical defenses of a new plant group, it promotes their diversification due to the availability of novel resources to radiate into. In this scenario, adaptation toward host plants is the driving force of insect species formation. And given our information in the quad, the two populations are geographically isolated, occur on different soil types at different altitudes, and possibly feed on different plants. One may wonder if the two populations have diverged, and if yes, to which point. This is a major preliminary question if we want to better protect the species. Indeed, we can face a challenge if there is a single taxon versus two taxa, which has implications for the conservation strategy. If there is a single taxon, then it is, for instance, possible that the two populations can interbreed and may increase species survival through positive heterosis, and this would require a more regional plan for the conservation. However, if there are two recognized taxa with substantial genetic divergence, for instance, it could be risky to interbreed the two populations and could lead to failure to rear the species due to mating incompatibilities. This, in turn, will lead to a more local conservation plan. So with this question in mind, we study the two populations of the quab through a set of specific work packages. I'm going to present briefly now this workflow. So briefly, it ranges from field sampling to molecular biology, analyzing the genome data, and also analyzing the transcriptome data. The genome is the total DNA of an organism, while the transcriptome is all the genes that are expressed to make the organism alive. Altogether, this is going to help understanding the species status and also the population trends of the quab to assist conservation. Thanks to the Swallowtail and Birdwing Butterfly Trust, we got funding to fly to Papua New Guinea and Pompondetta. I met Darren Bito there, who is setting up a rearing facility for birdwing butterflies and other butterflies. With Darren and his team, we went also in the field around Pompondetta and around Managalas Plateau to visit both localities of the two populations. In Afore, at the Dareki village, I met Conwell, who knows very well the quad how to find them, mostly the caterpillars actually. It remains difficult to see the adults because they fly too high in the canopy. So there in Papua New Guinea, I learned a lot of things, a perfect complement to me compared to the books and publications. So Darren has set up a rearing lab in Igaturu near Poponeta, where birdwings are reared, fed, and bred in fly cage. Here, as shown by the orange arrow, you can see a pupa of a quab that will soon emerge. However, for the sake of the project in questions, we had to sacrifice some quab specimens. I took a portable molecular lab with me to set up a small working station for preparing samples for DNA and RNA extractions. So we selected 2 specimens per population, with 1 male from the Managalas Plateau at the bottom left and 1 female from Popondetta at the bottom right. 2 caterpillars were also sampled, 1 from the Managalas Plateau and 1 from Popondetta. Each specimen was dissected into head, thorax, and abdomen and stored apart in RNA later for maximizing DNA and RNA preservation. We also took Ornithoptera priamus for comparison purpose, and we will see later why. Now, back in France, we started the sequencing of the quab populations in our lab. We sequence high-quality whole genomes for each specimen to make the most of these sacrifices. That will be reference genomes for this species. We used a combination of sequencing techniques with long DNA reads as performed in our lab with Oxford nanopore technology and short DNA reads performed externally by Novogenes with Illumina technology. In addition, we originally planned to sequence museum specimens using only Illumina technology in order to cover the geographic range and get better estimates of the genetic diversity within the quab. However, it turns out that there are issues with this because there are uncertainties about the exact location of capture from mixed specimens. Most of the specimens come from an area called the Kumusi River, which today is not known to house the quab, as you can see on the map on the right. And John Tennant has recently dig in mixed biography to identify this location. And he actually told us that mixed localities are difficult to understand. It was published with wrong data, but the nearest one can get to it is 2 days walk inland from the coast at Buna Bay, certainly flatland and certainly not at Biaggi, where the female holotype was claimed to be collected. Given this difficulty, we decided to put aside this part of the work. Let's proceed with the result of the genome sequencing of the quab. Thanks to the magic hands of Emmanuel, the sequencing went very well. Here I only present the result of the long read sequencing through Oxford nanopore technology for each quab specimen and one Priamus specimen. The genomes were assembled and analyzed by Elliott. You can see that we have obtained at least 66x of depth of coverage, meaning that we cover at least 66 times the whole genome, which is very high. We found a genome size of 321 megabases for the lowland population and of 325 megabases for the highland population. What is certainly of high quality here is represented by the N50 statistics, which define the assemblage quality in terms of contiguity. The N50 is defined as the sequence length of the shortest contig at 50% of the total genome length. Here, N50 range between 7 to 10 megabases, and we have assembled DNA contigs that are probably at the chromosome level, with the maximum length being at 13 megabases. Another statistic for the genome quality is the Busco analysis. Here is in the stacked histogram is represented the proportion of genes recovered out of a set of selected genes designed for all Lepidoptera. And the blue part represents the complete genes and is at least of 97% out of more than 5,000 genes in total. Now, let's compare our average genome to several Lepidoptera genomes for which we have good data. Here is a table presenting such data, and we have included the genome quality of the quab and Priamus. To me, the best genome is the Chinese peacock swallowtail, here in red, because they have used a combination of techniques that improve the genome assembly. But it is remarkable that the quab genome comes second in terms of quality, and this is astonishing given the rarity and difficulty to assemble and sequence non-model species. And we have 4 like this. So the genome of the quab will likely lay the foundations for exploring the genetic basis of biological feature of quab and for providing a useful data source for comparative genomics. Let's see that now. Thanks to this genome, we can estimate the mitochondrial diversity and also the nuclear heterozygosity. Here is provided an estimate of the mitochondrial diversity in mammals with orange and red bars showing endangered species and blue and green bars showing non-endangered species. Here is the same, but for nuclear heterozygosity. Well, human genetic diversity is about here, and a highly endangered species, the browniana, is here. Endangered species are commonly thought to have low mitochondrial diversity and low nuclear heterozygosity, which is often the case, but not always, as you can see with the chimpanzee. So now, what about the quab. Elliott analyzed the 4 mitochondrial genome of the quab and found a very low mitochondrial diversity with no more than 15 variable sites over more than 15,000 sites in total, which is about 0.1% of variation across all the mitogenome. This is lower than the polar bear or the gorilla, for instance. For insects, this is very low. A recent study in European butterflies shows that the intraspecific diversity is about 1.3% in average. And for swallowtail, you can see that some species have at least 8 times more mitochondrial diversity than the quab, even for some species that are considered as endangered, like Parnassus apollo. Now, what about a nuclear heterozygosity? Here is an updated plot of the genome-wide heterozygosity in mammals showing numerous species with different threat status based on IUCN. Elliott analyzed the quab genome and found a low nuclear heterozygosity with 0.086% of heterozygous sites. It's one heterozygous site every 1,200 base pair. This is at the same level than the Bengal tiger and lower than the giant panda or the Sumatran orangutan, for instance. Again, for insects, this is very low as compared to other butterflies and swallowtail. Just Priamus here on the top of the graphic is 4.8 times more heterozygous, and papilio species are at least 11 times more heterozygous than the crab. With the genome data, we can also estimate the demographic trends of the quab. Here, I show 2 examples. The brown hyena shows a continuous decrease in population size over time until the present. The vaquita, the smallest and rarest cetacean species with only 19 individuals alive today, shows another pattern quite unexpected for its status. There was a sharp population decline early in its history, followed by a stable but low population size since then. Of course, for other species, you can estimate expansion of population size toward the present. Now, what about the population trend of the quab? Elliott analyzed the 4 quab genome independently and found sharp decline starting 100,000 years ago. The effective population size dropped from more than 50,000 to less than 15,000, but at a slow pace, and it is stabilized over 15,000 since 11,000 years ago. Between 15 and 20,000 mature individuals for an insect species can be seen as small and reflect here potentially the restrained distribution range of the quab. It is important to underline there is a similar population trend between the lowland and highland population, here in lowland in blue, highland in green lines. Now we superimpose the estimate for the Priamus species. And we find that the population size also decreased sharply at about the same time than the quab decline. And there is also a recent shift at about 11,000 years ago. The difference is that the upturn is important so that the population size of Priamus increased to more than 300,000 mature individuals toward the present. The result also suggests the presence of a common factor leading to the population decline between 100 and 11,000 years ago. This, in fact, corresponds exactly to the last glacial period, with the last glacial maximum at 19,000 years ago, the bottom of the slopes here shown by the light blue arrow. The glacial period was a major upheaval even in the equatorial tropics, with contraction of rainforest and expansion of grasslands. It's possible the birdwings, as well as other species, did not benefit from this environmental change. Well, there is still work ahead before we can provide a definitive answer. With our genome data, we will assess the population structure to estimate whether there is gene flow between the 2 populations, and we expect there will be. We are also sequencing the RNA of the 4 specimens so that we can estimate the gene expression between the 2 populations, especially in relation to potentially different host plants. Finally, about host plants, we are sequencing the RNA to estimate the divergence between the lowland and highland population, and also to estimate the expressed genes for the toxicity. Altogether, we will have a better answer to the species status of the quad. In the meantime, we can still summarize this first result. We have obtained 4 reference genomes for the quad that tell us there is a single taxon with a very low mitochondrial genetic diversity and very low nuclear heterozygosity. This goes in favor of a regional action plan for the protection of this species. Altogether, this will mean that breeding the 2 populations could be advantageous. It's well possible there is a natural gene flow and thus natural hybridization in nature, and we will see this very soon. Well, I would like to thank all the people listed here, with a special thank to Elliot, who is doing an excellent work on the quad genomes, and Darren for the great help in Papua New Guinea and all the fieldwork we did. Finally, I would like to thank the SBBT for initiating the project and funding the travel to New Guinea. And now I would like to thank all of you who listened to this talk, and thank you for all this question.